Hello, everyone, and welcome to today's webinar, Talking Diagnostics, a Culture Moving to PCR. I'm Christy Jewell of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com. Now, we encourage you today to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. You may also submit any technical questions here as well if you have trouble seeing or hearing the presentation. And with that, I would now like to welcome today's speaker, Dr. Michael Perry, Deputy Lead Scientist, UK Anaerobe Reference Unit, Public Health, Wales Microbiology, Cardiff, University Hospital of Wales. For a complete biography on our speaker, please see the Presenter tab at the right hand of your screen. Dr. Perry, welcome. You may now begin your presentation.
Thank you so much for that introduction. It's a pleasure to be able to talk to you today about molecular enteric testing and our experience here in Public Health Wales and my experience over the last decade or more. So to start off with, just need to show this disclaimer from Thermo Fisher Scientific. Basically, you can blame me for whatever's on my slides. It is important for me to acknowledge my disclosures. I have done a lot of work over the last decade or more with molecular enteric PCR manufacturers, with other diagnostic companies, also with pharmaceutical companies and instrumentation companies. And the list of those that may cause some sort of issue with what I'm talking about today are included there at the bottom. So a little bit of background in relation to my employer, Public Health Wales. The organisation is part of the National Health Service here in Wales, and it includes multiple public health departments, including my own service, which is the microbiology service, but also screening, health protection, health and well-being, etc. Within microbiology services, we have a network of microbiology laboratories across the whole of the nation. Most of those are allied to Public Health Wales, and the aim of this microbiology service is to protect the public of Wales from infection. Also within the network, we have several reference facilities as well, and I work within the UK Anaerobe Reference Unit, which is located here in Cardiff in southeast of Wales, and there are other reference facilities as well, including, as I've mentioned here, the Cryptosporidium Reference Unit, as they were part of some of the work that we did in the evaluation and implementation that I'll show later on. A little bit more background in relation to the Anaerobe Reference Unit. We are based here, as I said, within Cardiff. We are sited in a hospital, the University Hospital of Wales, which is approximately 1,000-bed teaching hospital. We provide and have provided UK reference level services for the detection, identification and further characterization of anaerobes since the 1980s. As well as that, we provide a Clostridioides difficile typing service for the whole of Wales. And we have done for many, many years now. In fact, C. difficile ribotyping was developed here within the Anaerobe Reference Unit in the late 1990s, early 2000s. Since 2016, we have typed all GDH and PCR positive C. difficile samples. And we are very proud and fortunate that we have a UK accreditation service, accredited whole genome sequencing SNP typing service for C. difficile typing, which we have been live with now since 2020. And as you can see from the picture here in the bottom right-hand corner, C. diff is even written on our roads, or at least it used to be in this very dated picture now. To give you a little bit of background in relation to the experience that I have and that the organization has with enteric PCR, it goes back to the early 2010s when, first of all, we recognized that an area of our diagnostic laboratory which was particularly in need of modernization was the enteric lab. It came about fortuitously at the similar time as we were approached by the government to put in place some preparedness for the Olympics in 2012. As many of you may remember, the 2012 Olympics was based in London largely, here in the UK. But as part of the London 2012 Olympics, there were also some events and lots of athletes that were based here in Wales, which is only a couple of hours away from London. And so as a result of this, the government felt it important for us to have some rapid diagnostics in place, which included rapid diagnosis of diarrheal disease to help to prevent any outbreaks that could occur within that setting. Subsequent to that, we also had President Obama and many other leaders from across the world attend the NATO summit here in 2014 in Newport, which is again in Southeast Wales. And for that, the government also approached us to provide some testing for that as well in the event of any concerns. We then were part of some Welsh Government funding to continue to explore enteric PCR, and again had things in place for the Champions League final, the football final that was held here in Cardiff in 2017, with Cristiano Ronaldo in attendance at that match. And then subsequent to that, we received additional funding to enable us to not just provide this service when big events were in town, but to provide it to the population of Wales. And that led to a procurement, planning and implementation exercise that I led as the lead scientist in 2017 and 2018. So what did we identify back in the early 2010s when we first started thinking about enteric PCR? As many of you who have been or still are working with conventional methods, we recognized that the turnaround times were certainly not optimal. Also, the sensitivity and specificity of some of the tests that we were doing conventionally, culture, microscopy, also suffered for a lack of sensitivity and specificity. We had limited pathogen coverage due to labor-intensive and skill-rich processes, so we could only look for a certain number of pathogens. And there was a requirement sometimes to send multiple samples or to separate samples to go between different laboratories in virology, microbiology, parasitology, etc. And due to these numerous and somewhat fragmented processes, it was quite a confusing laboratory workflow, not just for sometimes people in the laboratory to make sure that samples went to the right place to get the right tests, but certainly for our users to understand how many samples they needed to send and what they needed to request on the different request forms. This is just some very early data back in 2011 when we started to think about enteric PCR and how that might impact what we saw in terms of our pathogen yield. And you can see there from the graph in the left-hand side that our pathogen yield was very low. In approximately 90% of cases, samples were negative for everything that we tested for. The potential benefits then of enteric testing were identified, included improving our turnaround times somewhat significantly for some of our targets, increasing our sensitivity, increasing and broadening our pathogen coverage, certainly improving our labor efficiency, making our processes much more lean than they were at that point, augmenting the clinical relevance of our testing in terms of our positive and negative predictive values, and enabling us potentially to look at syndromic testing using large multiplex methods, which I'll come back to a little bit later. So back in those early 2010s, we did a couple of evaluations that were assisted by some of the funding and time that were allowed in preparation for those big events that I mentioned previously. What you can see in the graph on the right there is a comparison of rates that we got in terms of our positivity rates for a host of pathogens. The blue bar is our prospective conventional work that we did, that we looked at, and then our retrospective conventional work in the red bar. The green bar is prospective nucleic acid amplification testing, and the purple bar is retrospective. And there's a couple of things just to mention there. We can see that in both of the NAT testing periods, both the retrospective testing that we did and the prospective testing, we saw an increase in our detection rates across the board. This clearly increased our negative predictive value. So giving out a negative result was a much more reliable result for our medics and for the patients. Particularly in relation to Clostridioides difficile, we saw that the sensitivity of detection was increased, as was the specificity. Previously, we were using a glutamate dehydrogenase assay, which detects non-toxigenic C. difficile. And so we were detecting more and detecting less non-toxigenic because the PCR targets are toxin targets, toxin gene targets at least. The other thing that we found here, looking at Clostridioides difficile, is that currently, certainly back in those days and still currently in the UK, the general algorithm is to look for C. difficile in all of our inpatients over the age of 2, and then only for certain populations outside of hospitals. With using a large multiplex where every sample was tested for C. difficile, we saw an increase in our positivity rate as a result of that increased access to that testing. Certainly that was the case for norovirus in our experience as well. Prior to introducing this, we were using a in-house molecular assay for norovirus, but the access to that test was very, very limited. And so increasing access not only increased the number of positives that we saw, but it also increased the sensitivity of detection going from our in-house to our multiplex method. The other thing that's really important to recognize, looking at that graph, as you can see quite plainly, especially from the norovirus detection, is that the prospective and retrospective periods that we used here were actually in different seasons. And so you can see that our prospective NAT testing was done in the winter when we see a whole lot more norovirus. So the next few slides that I'm going to show you outline some study data that we published in the last couple of years. The workflow that you can see there on the right-hand side just outlines what we were comparing in this study. We did not do a side-by-side validation or evaluation of these assays. The implementation would have been much too complex and costly had we introduced enteric PCR alongside our conventional testing. Because we had done all of that evaluation prior to implementation, we were confident what we were going to see. And so we had an implementation period where lab by lab, we went over to enteric PCR testing. So one day we were doing conventional testing, the next day we were offering enteric PCR. This was after a successful procurement in 2018, and also after national and local validation and verifications, which were undertaken over that 6-month implementation period. Something that we found was that the implementation went really, really successfully, as we had a lot of very enthusiastic and able registered and non-registered scientists involved from across the country. The other thing to point out here is the size of the numbers. So this is retrospective data downloaded from our laboratory information management systems. And what you can see is that we have a very, very large amount of data here, real world data. And you can also see that it's split into a stage 1 and stage 2 analysis. So stage 1 analysis is just looking at all patients and all samples, and stage 2 analysis is just looking at those samples where we had an idea of provenance, whether they were likely to be inpatients or outpatients at the time that they were tested. But we didn't lose too many samples in that stage 2 analysis, which was pleasing. What we can see in this slide is a representation of the methods that we were using prior to enteric PCR implementation, so the traditional diagnostic methods that we were using, and then also the gene targets that we were using post-implementation. It's important to understand in the slides that follow that any culture that we performed post-implementation was not for a confirmation of our PCR result. That was something that was quite difficult to get rid of, was this thought and vocabulary that we were doing culture for confirmation. That wasn't the case. We were doing culture for additional epidemiological information, such as susceptibility results, antimicrobial susceptibility test results. And so the testing that you'll see is not for confirmation, but for additional information. So in this slide here, you can see data that is comparing the results, the positivity that we saw in our nucleic acid amplification testing post-implementation with the percent positivity for those particular targets when we were performing traditional methods prior to enteric PCR implementation. And what is very clear there is that post-implementation across the board, both within inpatients and outpatients, we saw an increase in the number of samples that were positive for each one of our targets, each pathogen of interest. And that was for bacteria and parasites. We haven't included any norovirus information within this particular study, and so I won't talk about that any further. So again, reminding ourselves that the pre and post implementation culture, before enteric PCR implementation, we were culturing for all bacteria. Post enteric PCR implementation, we were only culturing samples that were positive. What we saw is that there was a reduction in our Campylobacter culture positivity rate. This is something that we had anticipated. We know that Campylobacter does not like to survive outside of the body and will often quite rapidly enter into a non-viable, non-cultural state. So it's important to remember that if you are doing culture post-enteric PCR, there will be at least 1 to 2, maybe more hours delay before you culture these specimens for Campylobacter and the other targets. So that is why our sort of hypothesis in relation to our reduced Campylobacter culture positivity. It's not something that we were overly concerned about because the information that we're deriving from our culture is mainly for epidemiological purposes. The fact that we are still culturing a very large number of our Campylobacter positive samples means that we are happy even with that slight reduction. It's noticeable that we also saw an increase in our Shigella culture positivity. We know that Shigella target for most, if not all, enteric PCR assays also detects enteroinvasive E. coli. Nevertheless, the big increase that we saw in this target cannot just be explained by detecting enteroinvasive E. coli that we weren't able to detect previously, because we can see that our Shigella rates have gone up, particularly in outpatients. And so using an enteric PCR method helps our scientists within the laboratory focus on those samples that are more likely to have Shigella, and so we're more easily able to recognize those colonies and report them and provide valuable information epidemiologically. We can see also that our E. coli 157 didn't really change, and the result of that is that obviously we still culture here in the lab only for E. coli 157, and I'll talk a little bit more about other sugar toxin, virotoxin producing E. coli detection in a minute. So the other interesting thing that we saw is that you were more likely to get a culture positive from our Campylobacter and our Salmonella outpatients than you were from an inpatient. This kind of turns on its head, I suppose, that hypothesis about the sort of viable, non-viable, and could suggest that it's actually more about patients who are being treated potentially, as we know that even though not many patients are treated for Campylobacter and Salmonella, more who are inpatients will have been because they'll have more severe disease. Again, it's not entirely clear, but interesting data. And we see quite a dramatic reduction in Shigella percentage positivity rate from culture in our outpatients. That may be reflected, a reflection of the fact that if you have Shigella compared to E. coli, enteroinvasive E. coli, that you're more likely to be hospitalized. And the converse again, looking at our sugar toxin or virotoxin producing E. colis, because most of those won't be E. coli 157. Most of those patients will be unwell, but not unwell enough to be admitted to hospital. As I promised, here is some of the data that we can see. So the vast increase in our positivity rate for sugar toxin producing E. coli is due to detection of additional sugar toxin producing E. coli strains, not just E. coli 157, which was all we were looking for previously in the main. And we can see that the vast majority as well of the samples were positive within the reference laboratory. This is some data on the parasites, again, just showing that when we sent these samples to the Cryptosporidium reference unit, that they were able, again in the main, to confirm, I've just used the word myself, our positives for Cryptosporidium and for Giardia. And then it just shows there the species or assemblages that were detected. Interesting that even though the PCR targets for the assay that we were using here were mainly Parvum and Hominis, we were still able to detect other Cryptosporidium species with our NAT assays. Finally, just looking at our interlab differences. So we have a number of laboratories that we did this testing in, and it's interesting to note that between those labs we had some quite different results, and that's due to the size of the lab, where they're located, whether they're more rural, and so there was quite a few differences. So if you do implement enteric PCR across a sort of network of laboratories, you will likely see similar differences than we saw. Just focusing a little bit more now on Clostridioides difficile. This is data from another publication from the last couple of years. And again, very healthy numbers in terms of the findings, I believe. So as I said, we transitioned from a GDH to a NAT test as our first line screen, maintaining the two-step algorithm of using a toxin EIA positive test as a reflex for all of our first screen positives. And you can see there that over the last few years, over the last 5 years or so, from 2015 to 2019, we saw a steady increase in the number of samples, the percentage of samples that were referred to the anaerobe reference unit. And so the data that I'm going to present now are really quite robust. Going from a GDH assay to a NAT assay, there was a lot of concern, angst about whether we were going to see a huge increase in our C. difficile numbers, our first screen C. difficile numbers at least, and potentially EIA positives as well. That is something that we just didn't see. In fact, we saw a decrease overall in our first screen positives. And when we delved into that, we found that that was because we were no longer detecting non-toxigenic or toxin variable strains, such as ribotype 039, 026. And you can see there that there was quite a stark difference. What we also saw as well is that looking at the labs that were still performing GDH testing, we could see that there was a huge variation in the isolation rate for C. difficile here within the anaerobe reference lab within those different GDH assays. So the labs that were performing GDH, one of them, we got an isolation rate of 56.3%, whereas at best it was 89.1%. And that really does show the vast difference in the quality of those different assays. And I would suggest that the lab that was getting the 56.3% that we were getting from our culture, from their samples, was using a very poor and somewhat inadequate GDH test. It was cross-reacting with things that weren't Clostridioides difficile. We saw a reduction in our overall toxin A and B EIA positivity rate. Numbers. The rate actually went up with NAT testing, but the overall numbers came down. So we were using a more specific assay for toxigenic C. difficile. And this was borne out looking at the ribotype profiles that we were able to before we moved over to whole genome sequencing. And you can see there that as we introduced NAT, that those toxin negative and a large proportion of our toxin variable strains disappeared or decreased. Just a few slides now talking about considerations for implementation if you're thinking of implementing enteric PCR. Things that you might want to consider from a laboratory perspective include the existing expertise or familiarity that your scientists have with PCR or nucleic acid amplification tests. This is something that will impact how fast you might be able to implement your assays, and also may also impact what type of assay you want to implement. Is it something that is going to involve and rely upon a high degree of molecular expertise, or is it an assay which is more suited to sort of less hands-on to allow people with less molecular expertise to make a start? Something that's important to consider is what sort of panel you want. Do you want a very large multiplex assay for a truly syndromic approach, or are you happier just looking for those targets that you currently look for with maybe one or two additionals? How much laboratory space do you have for your enteric PCR assays? Do you have more than one laboratory? Certainly if you're using some assays that may involve post-amplification processing, but even with any assay, it's always good to have at least two laboratories, one for sample preparation and setup, and then one for amplification. So have a look at your laboratory space and see what is feasible. For laboratory information management systems, you will almost certainly need to set up new test sets and consider whether you want to include CT values or CP values. And the bane of everyone's life, very often, interfacing. Get it started early is all I can say. It was a challenge. We went live prior to interfacing because it just took so much longer than it should have. Reimbursement is important for many laboratories, and so make sure that you want to be reimbursed for the assays that you're performing, and there will be problems if you're not able to. Think about outbreak response as well. How will your enteric PCR assays help you with outbreak response? I would suggest that they will be very helpful because they will give you much more rapid turnaround times. But once you're into a very large outbreak, there may be consideration as to whether you need to keep doing PCR or whether you can just go to culture or microscopy, depending on how you want to manage that outbreak. And last of all, just to reiterate, the positive vibes that we found really came to the labs as we introduced enteric PCR. It was a massive success and really filtered, well beyond the enteric lab in terms of the positivity that people felt as they took on new skills and introduced better assays. It's important, obviously, to consider the clinical impact, and that includes, again, existing expertise and familiarity. Are your clinicians familiar with PCR or NAT results? Is that something that is going to take a lot of education, or will they have transferable skills from interpreting other NAT assays such as respiratory assays, etc.? Again, it's important to talk to the clinical teams about exactly what they're looking for, especially in relation to reimbursement. If you look for a lot of things they don't want you to look for, they may not be willing to reimburse you. So make sure that any decisions that you make aren't just laboratory based, but they are in liaison with clinical teams. And most importantly, make sure that those decisions are mainly patient focused. Of course, we have things that we need to think about and consider from a clinical and laboratory workflow and expertise point of view, but ultimately we are providing services to patients, so let's put them first. Again, when you're reporting in LIMS, we need to think about how that will look to our clinical facing teams. Will they understand the results that we're reporting in relation to the CT values, whether we include those or not, whether that's important or confusing? And also, will these results interface with clinical systems, and what will they look like in those clinical systems? I've mentioned outbreak response and reimbursement, but again, just important to think about those from a clinical context as well, and make sure that your clinical colleagues are involved. So when you're thinking about the actual transition, so you've decided, yes, we're going to go for enteric PCR, what do we need to be thinking about? I would say first and foremost, especially because the landscape has changed so significantly since we started back in the early 2010s with a huge array of different assays and assay types available to you now, you need to, as much as you possibly can, understand what the current landscape is when you are looking to transition. And that will help to inform any procurement or tender process that you go through and make sure that you get the best that you can. Engage very early with industry partners. Invite them to come and talk to you about what they have to offer and explain to them what you're looking for, and that will help the procurement process to be a lot more smooth and to make that as efficient as possible. Wherever you can, evaluate assays so that you have a true understanding, not just of what you're being told, but of how these assays may be performing. They don't always do what they say on the tin. Make sure that you have a really good project team that understand why they're there, that have a range of expertise, that aren't necessarily all as optimistic and excited as you are. Make sure that there are some critical friends in there as well. Manage the expectations of that team and those that you are looking to implement enteric PCR for, and don't underestimate your timelines, especially, as I said, with interfacing. Early identification of stakeholders will help your implementation to be successful. And I've just put a small number of the important ones to include there. But et al. Is very important because that list is sizable when you actually sit down and start thinking about it. Make sure that you plan in time and cost for validation and verification work, and make sure that within the implementation you include again time and money for troubleshooting and then ongoing training and competency assessment post-implementation. Finally, just to talk about some of the benefits that you'll see from introducing enteric PCR. You will increase your pathogen detection accuracy, especially looking at key healthcare associated infections. You'll vastly improve your healthcare scientist efficiency and your patient and staff satisfaction. That's certainly what we found here in Wales. You'll reduce your turnaround times and positively introduce the production and disposal of hazardous waste. You'll also optimize the use of valuable resources, including staff, laboratory premises, and also hospital and clinic in terms of patient flow and journey. I'd like to finish just by acknowledging all of the colleagues that have made this presentation possible, particularly Trevor Morris, who is the lead scientist within the UKARU, and Dr. Harriet Hughes, who's the clinical director of the unit. Dr. Sally Corden and Fatima Downing, who were key in getting this work started and in the implementation. Fatima was the project manager. Lucy Holliday and Professor Lewis White, who were, Lucy was the main author of the study that you saw at the beginning, and Professor Lewis White was also another author. Dr. Robin Howe, who supported this work from start to finish, and all of our colleagues from Diagnostic and Reference Laboratories who contributed, and other laboratory partners such as Claire Jenkins in the UK HSA, or PHE as it used to be known, within the E. coli reference lab. And of course, all the industry partners that made this possible and have led us to where we are in offering a really valuable and improved service to that we were doing previously. And with that, I will thank you all for listening, diolch, as we say here in Wales, and I will turn the time back to the moderator and would welcome any questions.
Dr. Perry, thank you for that excellent presentation. It is now time for our live Q&A portion of our webinar. Now, just a reminder to our audience on how to submit questions. There's that Ask a Question box at the bottom of your screen. Please type in your question, click Submit, and we will answer as many of your questions as we have time for. Dr. Perry, we already have so many questions in the studio that have come in. I'm going to just go ahead and dive right in. First question. What is a handful of bacterial targets to test for bacterial gastroenteritis? Oh, Dr. Perry, I think you might be muted on your side. Let's see. I have you unmuted here. Not yet. It could be your headset. Let's see. Let me. Check your device settings real quick. Bear with us, everyone. We're going to get this.
Can you hear me now?
We can.
Okay, let me just. Yep, I can hear you now, so I'll just remove the headphones and hopefully it'll work.
Okay, perfect. Would you like me to repeat that question?
No. I think I remember it. So it will very much depend on where you are situated. I know that certainly within Europe, we have quite an array of different organisms that different countries are mandated to look for. I would imagine that most, if not all, laboratories will need to look for Salmonella, for Campylobacter, for Shigella. And then on top of those main bacteria, you may also need to look for virotoxigenic E. coli, especially E. coli 157. And then other places I know in Europe need to look for certain parasites and additional bacteria. But it really will depend on where you are and what your local epidemiology is and what the resource limitations are. For me, I would say definitely those 4 bacteria that I've already mentioned, and then at least Cryptosporidium and Giardia, maybe Entamoeba, depending on where you're situated. But those would be my top sort of bacteria and parasites.
Very good, thank you. Our next question. What do you recommend to effectively report C. diff positives?
So here in Europe and in the UK, we have a 2-step algorithm, which is really clinically beneficial. So we would have a high sensitivity screening assay, such as the glutamate dehydrogenase.
It appears we have lost Dr. Perry. Just a moment. Dr. Perry, can you hear us? Okay. Dr. Perry, real quick, we do have quite a few questions. Do you mind really quick refreshing your browser so we make sure we have a nice, clear connection? Thank you. And apologize for the technical difficulties, audience. We'll get this all set up real quick. And we thank you for your patience. I do want to remind everyone that any questions we don't have time to answer today, and those that are coming in during the on-demand period, they will be addressed via email, but we're going to try to answer some more questions here. Let's get Dr. Perry all connected.
Can you hear me again?
We can. Welcome back.
Thank you.
Very good. Do you mind starting from the beginning of that question, Dr. Perry, just so we make sure we get your full answer?
Yes, of course. So my preference would be a PCR test above a glutamate dehydrogenase test, whether that is part of a 2-step algorithm or a standalone test. Here in Europe, we have a 2-step algorithm, which is mandated, and I think that can be clinically useful. However, regardless of what test you use, you should be taking that test result also in light of the clinical findings with your patient as well. So regardless of what test you use, make sure you use it in combination with clinical findings, but certainly PCR is preferable to GDH for me, because you're not then confusing the clinical picture with toxigenic strains that are very unlikely to be causing C. diff, which is what you'll detect if you use a GDH assay.
Thank you. Our next question. What sample types are available for the NAAT and the PCR testing?
Sorry, could you repeat that one, Christy?
I can. What sample types are available for the NAAT and PCR testing for the NAT?
Yes. So here in Wales, we just use pure stool, diarrheal stool. I am aware that some assays these days do have findings for swabs, and obviously I think in the US their Kerry-Blair medium is quite popular as well. So there will be a lot of assays that have clearance for fresh stool, for stool in Kerry-Blair medium, and now some for swabs as well, so for rectal swabs. But here in Wales, we use just the fresh stool without any transport medium. Obviously, it's important that samples get to the laboratory as soon as possible, and ideally are refrigerated on the way. I'm quite keen on rectal swabs. I think that could be quite a saving in terms of the planet, in terms of transportation and cost, etc. In the future. So I would very much be keen to explore rectal swabs a bit more here in the UK and across the world. But currently, it's fresh fecal specimens.
Thank you, Dr. Perry. Next question. When should I have targeted approach? 1 to 4 targets versus a real syndromic approach, 10 to 20 targets?
That's a really, really good question. And I suppose it relies upon a few different things. One is, what your customers are willing to reimburse you for. What do they need? People who are immunosuppressed, there may be a desire to test for a wider range of targets, targets that maybe don't cause diarrhea as often in immune competent patients. So you could choose to use a wider panel for particular patient populations. My personal feeling is that the syndromic approach is very meaningful. And something that I would like to head for, in the future would be a larger array of targets. But I'm mindful that you have to balance that with the cost of testing as well. So I would say at the very least, if you introduce enteric PCR, you need to be covering the targets that you had previously with your conventional methods. Obviously, anything additional will, I think, help your business case. And improve the customer satisfaction, and ultimately working towards these larger syndromic panels, I think makes a lot of sense, because at the end of the day, diarrhea is diarrhea, and it's very, very difficult to say what is and what isn't causing that diarrhea until you actually have a laboratory result. So yeah, it's complex, and it's something that has to be decided on a case-by-case basis within your institution. And within your sort of area.
Thank you, Dr. Perry. And it looks like we have time for one more question, but I want to, before I ask, direct our audience to a survey question that we do have for them as well. If you look in the bottom left corner of your screen, you'll see a survey question. We'd love your input. So Dr. Perry, our last question, how long would it take to train a lab team for enteric PCR if they have been doing culture and microscopy for long?
Yeah, really, really good question. And again, I'm going to answer it in a slightly woolly way. That does depend. There are people who pick up molecular diagnostics very, very quickly and embrace it. There are others who, for whatever reason, and it doesn't seem to be, age specific at all. There are people in the lab who just enjoy conventional techniques. They enjoy sort of the artistry that comes with them. And so often those members of staff can be somewhat resistant and take longer to train. I would say if someone, is embracing the technique, between 1 to 3 months, certainly, within 3 months, you can get them up to a good level. Again, that's something to consider when you're looking at what assay you want to introduce. If you feel that your laboratory team may be a little bit resistant or less able to pick up the skills as quickly, then you might want to look at a more automated type method. But certainly we found that non-registered staff embrace these techniques very, very well. And most within a month to 6 weeks really were very, very skilled. Certainly in the practical side of things. The analysis is a little bit different. And again, that's something to consider when you're looking at different assays, is how much sort of analytical work is required. Is analysis completely automated? And so that takes the pressure off laboratory staff to sort of learn those analytical skills, or is it something that they will need to learn? With a molecular diagnostics background myself, I think it's very valuable for scientists to have those skills. But yeah, again, it will be specific to your laboratory team and what you feel is best suited to them. So hopefully that sort of answers the question in a way that will help most people, no matter where they're coming from.
Thank you, Dr. Perry.
One thing I will add to that, actually, Christy, the other thing that I do feel, even as a molecular scientist, I think it's really important that we maintain those conventional method skills as well within our laboratory workforce. And so I certainly would not be in favor of getting rid of culture and microscopy and other techniques completely. I think we sort of saw that a little bit with in virology, when we went from testing respiratory viruses in the main using cell culture to molecular. Here in the UK, at least, the skills for cell culture almost disappeared overnight. And I think it would be a real loss if that happened in bacteriology as well. So I would certainly say, no matter how skilled your workforce becomes in molecular techniques, make sure that they have the opportunity to maintain their expertise in conventional methods, because they are really important. And of course, when it comes to pathogens that maybe are new or novel, that we might not be able to get a molecular assay for as quickly as we might like, we may have to revert to microscopy or culture, sort of very basic means to detect those in the future. So I think it's really valuable to maintain both molecular and conventional skills.
Dr. Perry, thank you so much for your important information, your research, and your valuable insight. I do want to thank Labroots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Now, before we go, I do want to thank our audience also for joining us today and for their interesting questions. We had several questions we weren't able to answer, and those that are submitted during the on-demand period, they will be addressed by our speaker via the contact information you provided at the time of registration. Today's webcast can be viewed on demand, and Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now. Thank you so much for joining us. Thank you again, Dr. Perry, and everyone have a great day.
You're welcome.
Bye-bye.
Bye-bye.